Hi, everybody, and welcome back to Miss Angler's biology class. I am Miss Angler, and in today's video, we are going to be looking at microorganism diversity. Now, this is an introductory video, so I'm going to be covering the basics around viruses, bacteria, protists, and fungi, their structures and how to identify them, and the diseases that they cause. For more specific cycles like how viruses reproduce or osmoregulation in protists, those can be found in the videos linked above. Now, the first defining thing that we need to look at before we go into the diversity of microorganisms is prokaryotes versus eukaryotes. And this is referring to their internal structure of the cells and how we tell the difference. So importantly, the names give us a clue. A prokaryote, often we refer to pro being primitive or first, kary referring to the nucleus. So it has a primitive nucleus or primitive organelles versus a eukaryote, which refers to a more modern nucleus or a set of organelles surrounded with a membrane. Now, what's important to look out for is they have some similarities between them, the two sets of cells, but they also have a few differences. Now, the three things that these eukaryote cells and prokaryote cells share is they both have some form of DNA, they both have ribosomes for protein synthesis, and they both have a cell membrane because that's a key feature of being a cell in the first place. But the most important thing that is different between the two of them is the presence and absence of what we call a true nucleus. If we look at the eukaryotic cell on the left-hand side here, we will see that it says it has a membrane enclosed nucleus. And as you can see, the nucleus itself is encased in a membrane. So it is surrounded by a protective layer. Versus if we look at the prokaryotic cell, on the other hand, you will notice that the DNA is freely floating in what we call a nucleoid, which is to some extent an advantage for some things, but it's also a disadvantage because it's not protected by anything. The next defining thing is those other organelles that are not bound by membranes that we see in the prokaryote. You'll notice in the prokaryote, if you look inside of it, you don't see many other organelles other than perhaps a ribosome here or there. And if this was a photosynthesizing bacteria, maybe a chloroplast or two. But other than those, you do not see endoplasmic reticulums. You do not see many mitochondria. You do not see any of these membrane-bound organelles that you see in eukaryotes. And so if you look at the eukaryotic image, you can see that there are membrane-bound organelles like the mitochondria up at the top here. We've got some Golgi body over here. We've got the endoplasmic reticulum. These are all membrane-bound organelles. Now, in exams and tests, you will be asked to differentiate the two, list the differences or tabulate them, and you need to have a clear understanding also of how to identify them in pictures. So let's look at our first group, viruses. Now, viruses are an interesting group because technically they are not cells, they're not alive, and so we refer them as acellular organisms, and viruses are considered to be acellular because they don't have any organelles, they have no nucleus, and they have no cytoplasm, and they don't carry out the 7 basic life functions like reproduction, respiration, excretion, digestion, etc. The only way for them to exist is within a host, and that host allows them to reproduce. Now, when studying viruses, we need to be aware of some characteristics that they have, starting off with their shape. And generally, viruses come in either a spherical shape, like this one over here, or they come in a more rod-like shape, like this one here. And these are two classic examples. One is the influenza virus, and the other is the bacteriophage. Now, in exams and tests, you will probably be asked to draw or label a virus. And so that means that you are going to need to be able to identify certain things and also know roughly what they do. Now, what makes viruses unique is they have a structure called a capsid. Now, a capsid is a protein coat that protects our virus, and it is present on this one over here. And you can see it's this layer that sits around the internal structures of our virus, where all our DNA or RNA is. And then on the outside of that, we also find some viruses have an envelope, which is another protective layer. So viruses often have a double protective layer, and that allows them to exist inside and outside of their host with ease. And that's also why it's really difficult to kill viruses, because of these two protective layers. Now, what you'll notice between the virus for influenza and the virus for the bacteriophage is they both have some kind of nucleic acid. The one has RNA, the other one has DNA, and that is the most important part of the virus because that's what we use to make more viruses. That's what they're trying to do. Now, if you're looking for virus reproduction, that will be in another video. So if you want to look for the link for that one, you can go ahead and look at my playlist, or I'm going to link it above. Now, when drawing a bacteriophage, it's got a couple of little extra pieces. It's got the head at the top, which is very self-explanatory. It's the top part, the neck and the tail. And essentially what happens is this bacteriophage will attach itself to a bacterial cell. That's who it infects. And it sticks with these tail fibers. It actually sticks to the cell. And it injects its DNA through the neck, down the tail, and into the bacterial cell where it goes on for replication. Now, most viruses do cause different diseases, and so we often refer to them as being pathogenic. And they cause many fatal diseases. In other words, you can die from a viral infection. Things like polio, HIV, foot and mouth disease, these are all examples of diseases. The next group is bacteria. Now, bacteria are one step up from viruses, and that's because they are living and they're a little bit more complex. And so we refer to them as being unicellular, which means that they either exist as a single cell or many of those single cells live in a colony together. And those colonies can be millions and billions of bacteria all living together. Now, when we categorize bacteria and we classify them, we often look at their shape, which is really important. And so there are three main shapes that we look out for, and they are cocci, bacilli, and then we get what we call others like vibros. And basically, the name is linked to their shape. So cocci is circular, bacilli is a rod shape, and then vibros is a sort of rod with like a tail. And there are many other options where you can have many of the cocci stuck together. Like you can see there's something called streptococci, so it's a whole bunch of them joined together. Same for bacilli, you can have many attached to one another, and that's how we classify them based on their shapes. Now, when it comes to exams and tests, you are going to be asked to either draw or label a bacterial cell, and you're going to need to know the functions of those particular structures. So we start off with the capsule. Now, the capsule is a protective outer layer, very, very similar to the one that we see in viruses. It's there as a mechanism for protection, along with the cell wall. And yes, the cell wall is exactly as we see it in plants. It's very similar in its structure, and it's the same in its purpose. It's there for rigidity, but also for protection. And of course, it has a cell membrane or a plasma membrane, which means that it is a cell. Remember, the virus doesn't have a plasma membrane, therefore it's not a cell. Now, some things that are different, and this is why bacteria fall in their own category, which we described at the beginning of the video, which is eukaryotes, is that their internal structures are missing a few things. They don't have membrane-bound organelles. So, for example, if we look on the inside, they do have cytoplasm, but you'll notice that their DNA, also known as their nucleoid, is freely floating on the inside. It's a long, like spaghetti string that isn't attached to anything. It's not inside a nucleus. You will notice that there are some ribosomes, but other than that, you don't see any mitochondria, you don't see any Golgi body or endoplasmic reticulum, and that's because bacteria are eukaryotic. What they do have, which makes them very unique, and that's why I'm going to highlight them in a different color, is they have structures called plasmids. Now, plasmids are really important because they allow bacteria to undergo replication a lot faster, and they also allow them to adapt to their environment. And we'll talk about that in another video about why the plasmid is so important. Now, something else bacteria have that's unique is they have flagella or flagellum or flagelli. And basically that refers to tails. So flagelli is multiple and flagellum is one tail. And they allow the bacteria to swim around and to move. And most bacteria is found in some kind of like medium, like liquid, but bacteria can also move through the air. And finally, you have something called the pilus. Now, the pilus are these like little pointy sort of spikes that stick out of the bacteria, and they help the bacteria to stick onto the organism they're trying to affect, especially if they are pathogenic bacteria and they are the ones causing diseases. The next group is Protista. Now, Protista are a very unusual group because, as you'll notice, they seem to share characteristics with everybody. And this picture here looks very similar to the bacterial cell image that we just looked at now. The shape is similar. There's a tail, that flagellum is there. But I want you to know that there are some defining things to look out for to be able to tell the difference when you see these in tests and exams. Now, the first thing we need to talk about is how they exist in the world. And they can either be unicellular, which means they are one cell, they live on their own, often in a body of water like a lake or an ocean or a pond. But they can also be multicellular, and they live as one multicellular organism. An example of that is something like seaweed. Seaweed's not a plant, it's a protist, but it's multicellular. It means it has many, many, many cells making up the protist as a whole. Now, when we are preparing for our exams and our tests, we need to know the structures and their functions. And I'm going to highlight some of the common things that we see, but then I'm going to point out what makes a protist a protist. Now, some of the things that we are looking for is the nucleus. So the nucleus is present in a protist because it's a eukaryote. Remember, it has membrane-bound organelles, and sitting inside the nucleus is the nucleolus. We should be familiar with these. You'll also notice that there are chloroplasts, and often Protista are photosynthetic, which means they can photosynthesize, and the majority of the plankton in the ocean are protists, and they photosynthesize. Now, the things that make protists unique is this label over here, the contractile vacuole. And this is a very unique kind of vacuole because this vacuole can appear and disappear, and its purpose is to regulate the amount of water in a protist. Why do you need to regulate it? Because protists are found in the ocean. They're found in lakes. And so they have to regulate how much water they absorb. If they don't, they actually end up drowning. So the contractile vacuole is unique to protists, and you're not going to see these in bacterial cells. What you will see, which is a bit of an overlap, is you'll see the flagellum, which is a tail. And this little flagellum is often one, maybe it's multiples. This is an overlap with bacteria. So you may see this in other diagrams. Now, a label that we don't see here, but I want you to take note of this because they do exist, is there might be another little vacuole in here. There might be a little circular structure here, maybe another one over there. And what those are food vacuoles. Now, food vacuoles are places where we store food until we are ready to digest them. Now, that means that protists have the ability to store food until they're ready to digest them. They don't have a digestive system like a stomach and an esophagus and intestines. So what they do is they surround their food with what we call pseudopodia, false feet, and they digest their food by turning it into a food vacuole. Now, if you're looking for the video on pseudopodia and osmoregulation, so how protists regulate their water, you're going to be able to click the video in my playlist, which is called Osmoregulation in Protists, or you can click the link above now. Now, the final group that we are looking at is fungi. Now, fungi can be both unicellular or they can be multicellular. If they're unicellular, they're going to be microscopic. If they're multicellular, they're going to be macroscopic, and you can see them with the naked eye. Now, what makes fungi so interesting is that fungi often were misidentified as plants for a very long time. But what we found is that fungi cannot photosynthesize, and instead they actually absorb their nutrients out of the substance they're growing on. And so we call them saprophytic, which basically means they sap or they suck out the nutrients from the thing that they're growing on. Maybe it's a dead animal, maybe it's a piece of cheese in the refrigerator where you get mold growing on the cheese, but it's something where they're taking the nutrients out of that thing. And fungi only grows on animals and plants that are dead or currently dying. Now, fungi comes in both uni and multicellular, but I'm going to focus now on the multicellular version. And this diagram here is of bread mold. Yet again, you may be asked to identify fungi and be able to label it and give some functions. Now, fungi is eukaryotic, which means it does have a nucleus with all of its organelles, so it is quite complex. It doesn't have any chloroplasts inside of it, so they can't photosynthesize. And instead, they have a lot of other functions and a lot of structures that help them live their lives a lot better. And so the first thing we're going to look at is the lower portion of the fungi, and we're going to talk about these substances called hyphae and rhizoids. Now, rhizoids are the equivalent of roots. They're not actually roots. They're similar, but basically they use them to absorb nutrients. Hyphae, on the other hand, are these long, elongated sections of the rhizoids. And this is what sometimes becomes quite confusing. Hyphae are basically long cells. So if I just sketch one here, they're a really long cell. But what's interesting is they don't have an end. Normally there'd be like an end to the cell here. Instead, there's just another cell that is right up against it with no cell wall. And the reason for that is that it allows for nutrients to like freely move through the cells very, very easily. Now, another kind of, let's call it thread-like structure that we see is a stolon. Stolon are basically hyphae that are, or rhizoids that are growing sideways. So these are the ones that we see on the surface, whereas rhizoids are the ones that are in the substance. So you can't actually see these, they'll be attached. Together, if we take the stolon, and we take the hyphae and we take the rhizoids and we put them all together, we form the mycelium. So essentially, mycelium is the entire body of fungi that we find below the substrate. In other words, what's growing inside the bread, or the cheese, or maybe even on the surface of maybe an animal that's died. Now, everything above that is not necessarily used for absorbing, but for reproduction. So what you'll notice is we have something called a sporangiophore. And a sporangiophore is referring to this structure, this long structure over here, where we are going to reproduce from. And at the end, there's a ball-like structure that's called the sporangium. That's the whole round structure. And each one of those tiny little round circles is a spore. And when those burst or they fall off, as you can see here on the side here, there's little spores coming off. They go off, they land in another location. Maybe it's further on a piece of bread. Maybe it is on another part of the animal that's dying and decaying. And those spores will grow into rhizoids and hyphae, and then slowly but surely they'll grow another sporangiophore, and they'll do this all over again. And what they are doing is fungi is reproducing through spores so that they can spread out like a mat, like a big web, grow over everything, which I'm sure you've seen like mold grow over like cheese and bread. It spreads over the whole thing. And how it does that is with those spores. Now, as always, at the end of every lesson, I like to wrap up with a terminology recap. So let's take a look at all the words that we've done, and you can use these to create flashcards. Now, we have prokaryotes and we have eukaryotes, which represent the two kinds of cells we get that have either membrane-bound organelles or they don't. We have viruses, which are acellular organisms, which are non-living. And that basically means that they don't have the ability to live without a host. Then they have two protective layers, a capsid and an envelope. A capsid is a protein layer and an envelope is a fatty layer, both of them allowing viruses to live anywhere. We also learned the word pathogenic, which means it causes disease. And a lot of viruses and bacteria and fungi and protists are pathogenic. They cause diseases. We then looked at bacteria, which are unicellular, which means they live on their own as a single cell, but sometimes in colonies, and they come in different shapes. Coccus, which is circular, bacillus, which is a rod, and spirillum, which is a spiral. Now, you get things called flagella, which are those long tail-like structures that allow bacteria to swim around and move. We then have something called pili, which are those spike proteins that stick out of a bacteria, allowing it to stick onto its host. for whoever it's going to live in. Then we have a plasmid, which is a circular ring of DNA. It basically is a special piece of DNA that allows a bacteria to reproduce. Then we have Protista. Now, Protista are these unusual microorganisms that live in water, and they have two very special vacuoles. They have a contractile vacuole, which moves water around inside of them, but they also have a food vacuole, which is where they digest what they have ingested. And they also have something called pseudopodia, which are false feet. I'm going to do a whole video on pseudopodia formation and contractile vacuoles as well. Then we looked at fungi. Now, fungi are saprophytic. It means that they break down dead and decaying matter, and they absorb the nutrients through their rhizoids. And then they have hyphae. Now, hyphae are cells which are open at the end. It means they don't have any cell walls and they grow underground. And then you get a stolon. A stolon is a horizontal hyphae. It means it grows above the substrate, and it is the thing that's growing like horizontally. Now, as always, if you like this video, don't forget to give it a thumbs up and subscribe. I post new videos every Tuesday and Thursday, and I will see you all again soon. Bye.